Good morning. Thank you for joining us on this cold morning. This lecture is to honor the memory of Dr. Stephen Straus. Steve was a distinguished scientist and physician and the first director of the National Center for Complementary and Alternative Medicine. I have the honor of being his successor as the director of that center. Steve's being honored twice in the current time at the NIH. There was a lecture last week to remember his exceptional contributions to infectious disease, his contributions as a distinguished virologist, and his contributions to vaccine development. These were extraordinary accomplishments. But today we honor and remember some contributions of a rather different sort. When Steve assumed leadership of the National Center for Complementary and Alternative Medicine, this was actually a rather brave undertaking. NCAM was created because of strong public interest in an extraordinarily wide range of health promotion activities. There was little acceptance in mainstream medicine that there was much value in studying these practices, and little scientific consensus on how to do it. Much skepticism on all sides about the value of the enterprise. I first encountered Steve when we worked together on the development of a placebo conference. It was there that I met recognized his prodigious intellect and exceptional imagination. He recognized that the placebo effect was of interest not just because it was a nuisance in clinical trials, but that it was a window on the incredible mind-body interface and the impact of context, reassurance, and expectation on health outcomes. This has remained one of the very fruitful areas in NCAM's portfolio. The principles Steve established for NCAM to bring rigorous science to a variety of healthcare practices, coupled with a good dose of open-mindedness and willingness to explore unusual avenues, has, I believe, stood us well. I think about Steve often. I hope that he would be pleased to see what we are doing with the foundations he built. And I'm pleased today that we're able to get together to honor his memory. It is my honor also today to introduce and welcome Dr. Vikas Sukhatme. Vikas's lecture is titled Promise for the Future in Yesterday's Remedies, Traditional Therapies to Modern Medicine. Vikas is Victor J. Aresti Professor of Medicine at Harvard Medical School and Chief Academic Officer at Beth Israel Deaconess Medical Center. He's Chief of the Division of Interdisciplinary Medicine and Biotechnology at Beth Israel. He has a Ph.D. In theoretical physics at MIT. I think he was about 19 when he completed that, and then went on to do an MD degree and clinical training in internal medicine and nephrology. His imaginative science has always excited my colleagues in nephrology. He has a strong interest in tumor metabolism, tumor immunology, and is co-founder of an organization called Global Cures, which is conducting clinical trials on promising therapies that are not pursued by the for-profit sector. In today's talk, he will explore with us promising and affordable therapies from outside the mainstream. Please welcome Dr. Sukhatme.
Thank you very much, Dr. Briggs. It's a real pleasure to be here. Thank you also for that very kind introduction. I didn't have the pleasure of meeting Dr. Straus, but was able to see some of the videotapes from your archives. And it's clear that he was a gentleman with very wide interests and a very keen intellect. And so it's a real honor to be presenting this in his memory. Let me start with just a couple of words of introduction. Since Dr. Briggs had something to do with choosing this title, I thought I would take the prerogative of modifying the word traditional just a bit. As many of you know, I don't consider myself a practitioner of traditional medicine as it's typically defined, nor do I study traditional medicines, if you will, at the bench. So I'm going to use the word traditional to refer to ideas, therapies that are old, that have been around for a while, but which have, for a number of reasons, not found their way into, if you wish to call them, mainstream medicine. Second point I wish to make is that the talk, despite its title, will be largely focused in the area of cancer. That's the area I'm most familiar with. But I will have some comments to make towards the last third of this talk that will go well beyond the issues of cancer. So let's begin. We'll start with some stories. The story goes back to the early part of the 20th century. A woman by the name of Joanna Brandt claimed in South Africa that she cured herself of her gastric cancer by a diet that consisted of alternating periods of starvation with eating for 12 hours, sipping on this mush of grapes. The seeds, the exterior, the skins, and everything in the middle. Now, there's no clear documentation of this in terms of a biopsy, etc. She did, in fact, have gastric cancer. There are a few anecdotal reports one finds on the web. But she wrote this book that's shown here on the left. That was the cancer cure using grapes. Here's another idea that's been around. Turns out another Joanna Budwig, a German biochemist who lived for about 100 years, actually, spanning, in fact, the 20th century, claimed that this diet, a diet of what were called quarks at the time, interpretation in today's day is cottage cheese, and a lot of flaxseed oil, tablespoons per day, it turns out, she said was able to cure cancer. And she provided, again, a number of data from a number of patients that she treated, and there's lots of these reported on the web. So we'll come back to this in a few minutes. The third story is of a very different kind. Gentleman, a surgeon, Harvard, Yale trained, who, as he started his practice of medicine at what is now the Memorial Sloan Kettering Institute, Cancer Center, came across some interesting observations. He was very dissatisfied as a surgeon in terms of the care he was providing to his cancer patients. He had noted some old observations that certain patients with cancer, if they had survived a serious infection, occasionally their cancer would go into remission. Now we're talking about cases of metastatic disease. This observation goes back, in fact, to the 1800s. Few patients were noted in the literature. And he, in particular, had seen a case where a person came down with erysipelas. As I'm sure many of you know, this is a dermal infection, typically caused by strep pyogenes. Occurs in infants, occurs in adults, sometimes originates because of a scratch, a wound. And he found that this particular patient with sarcoma had had a complete remission following this infection. This is, of course, pre-antibiotic days. So this led him to think about the notion of actually intentionally injecting bacteria into a cancer patient. And over a period of several years, he decided that this mixture, strep pyogenes and Serratia marcescens, was the mixture that he would do most of his work with. Initially started off with live bacteria, later on proceeded in what were then called the Coley's toxins to a killed mixture. And in general, if you look over the decades of work that he did, he found that repeated intratumoral injections to create fevers to 102, 103 and above, and higher seemed to work the best. He documented his treatment in over 900 patients over several decades. And I'll show you that data summarized in the next slide, with about a 50% 5-year survival rate. Quite dramatic. And what was quite curious which brought him a lot of attention but also an equal number of detractors, was that there was efficacy in a broad range of tumor types. Epithelial tumors, connective tissue tumors, were all on this list. So take a look here. This is divided into two groups, so-called inoperable tumors and operable ones. Approximately a 50% rate for a 5-year survival across a whole range of tumors. Now, we'll come back to this also. I've laid you out 3 stories, all from yesteryear. Now, obviously, cancer has not been cured. Obviously, these therapies have not become mainstream. In fact, not only has cancer not been cured, but it's quite clear that the burden of this disease at the moment is still enormous. So you would think, based on what I have said, that the logical conclusion would be that we should leave no stone unturned. If we need to, we should think out of the box. So there's the proverbial box, and let's start thinking outside of this box today. So the talk I will divide into 4 parts. You've heard parts of the first one. I will try to suggest to you that these 2 areas, which are by no means mainstream oncology, in fact, the word tumor metabolism, for example, is not mentioned in Bob Weinberg's book on cancer. So this is not a popular area. And this area, that of tumor immunology, I believe is now on the rise, as in fact is the attention to tumor metabolism, for that matter, but also is not one that has proved very successful in the current treatment of metastatic disease, with some notable exceptions in renal cancer, perhaps a little bit in melanoma, and more recently in prostate cancer. In solid tumors, we have not seen yet the impact, significant impact, of work in tumor immunotherapy. I will then tell you that a lot of what goes on in these 2 brackets, where mechanisms may not be perfectly defined, where we may not even know the active ingredient or ingredients, it'll be important as we think of making those studies rigorous to truly rely on imaginative and sophisticated biomarkers to guide our way. And then in the final part of this talk, this is sort of all science, if you will, what gives? If the science, in fact, does turn out to be solid, will these therapies inevitably move into the clinic or not? That's part 4. Okay, so I'll pick up the pace now a little bit. So here's a landscape of cancer therapy. I'm talking about metastatic disease now, so surgery is not included on this slide as a therapeutic regimen. Most of the attention, even in metastatic disease, the attention is focused on activity that, or therapies that act in the vicinity of the tumor, primary tumor and at the metastatic sites. So the tumor cell is an attention, of course, and the non-tumor cell is a target. And cytotoxic, differentiated therapies of the type that we're talking about here, of course, affect the tumor cell. In some cases, these also will affect the non-tumor cell compartment. Anti-angiogenic therapies affect blood vessels, immunotherapy, immune cells, and so on. There's not much that we're targeting towards fibroblasts, for example. But we now know full well that these 2 act in concert. And there's some very nice descriptions of this in a nice review by Weinberg and Hanahan, which points to the close interrelationships of these 2 cell types. That is, that the tumor no longer should be regarded as an entity that is composed solely of tumor cells. There's quite an interesting synergism and crosstalk that occurs between the tumor and the non-tumor cells in the environment. Acellular elements, the matrix that tumor cells reside in and so on, is of course another area of considerable interest that is picking up. Now, not as much attention has gone on to what's going on outside of where the tumor or tumors primarily reside. In other words, we know full well what is the obvious cancer phenotype that we see in a person. The person is emaciated. Some cancers more than others. There's clearly a crosstalk going on here between the tumor and a whole bunch of other cell types. What's going on in fat cells? What's going on in breakdown of muscle? What is the liver's role in all of this? What are all those connections? Very, very poorly understood. Some amount of work has gone on in this subject. What is the mechanism of cachexia is what I'm getting at. But very little has really come forward in that. So the word metabolism or metabolic therapies doesn't figure on this chart. But as you'll see in just a few minutes, I think there are some very interesting insights that metabolism can guide us towards both the tumor cell compartment, the non-tumor compartment, and outside the tumor as well. And I'll talk about that in just a second. So let's move forward. Here's a timeline of discoveries in the field that I'm calling tumor metabolism. No discussion of this field can omit the Warburg effect, and so I'll spend about 10 minutes on that. The idea which occurred in the early part of the 20th century. Glutamine as a fuel, not recognized till about 60, 70 years ago. And that is, we all know we have glutamine in many of the media that we use to grow tumor cells. The fact that there are specific isoforms of enzymes in metabolic pathways that are specific for tumor versus non-tumor cells has been recognized over the last 20, 30 years, and more recently in an accelerating way. The idea that oncogenes and tumor suppressor genes, which of course are the genetic basis for what gives rise to cancer, the fact that these drive changes in tumor metabolism has been relatively recently appreciated, and I'll show you some examples on that. And this idea that metabolic pathways and our ability to intervene in these pathways actually talks back, if you will, to signaling pathways is, I think, quite new. And I'll show you some data that we have in our lab to support one example of that. So let's start with the Warburg effect. Otto Warburg, 1883, 1970, Nobel Prize, though actually not for the Warburg effect, for something else, but described this phenomenon. The phenomena is as follows, that in general, in normal cells, most glucose that is taken up by the cell is converted to pyruvate. And that roughly for every 100 molecules of pyruvate generated, 5, 10, 15% might go to lactate. The remaining feed into the Krebs cycle, pyruvate converting to acetyl-CoA, feeding into the Krebs cycle, and then the byproducts of the Krebs cycle, NADH and FADH2, going into oxidative phosphorylation and ATP being generated. You can get a lot more ATP generated if you go down here than if you go down here. Here you get 2 molecules for every molecule of glucose going down. Here you get 30 odd. And I say 30 odd because there's some interesting shuttles that come into play that I don't want to get into, which define exactly how many that happens depending on which shuttle pathway is used. Now, what Warburg discovered is that that ratio is flipped in many cancer cells, in the majority of cancer cells. And it's flipped even in the presence of adequate O2. So this phenomenon is sometimes referred to as aerobic glycolysis. Glycolysis is, of course, the conversion of glucose to pyruvate. And so even under aerobic conditions, you have more pyruvate going to lactate than typically would occur in a non-cancer cell. It might be 50-50. It's not typically 95-5, but 50-50 is quite different from 5 or 10 versus 90 or 95. That's what Warburg discovered. So let's ask a number of questions. How does the cancer cell benefit from the Warburg effect? Now, there are no simple answers to this, and there are a number of answers. I'll give you some. What I didn't show you on the previous slide is that the first step in glycolysis is the conversion of glucose to glucose 6-phosphate. There's a pathway that comes off from glucose 6-phosphate that does not go down into glycolysis, and that is the pentose phosphate pathway. And this pathway generates certain nucleotides and generates NADPH, a key reducing molecule. Another offshoot further down in glycolysis will generate glycerol, the backbone for the fatty acids that will form, that come off from citrates. So citrates is part of the TCA cycle, and there is in the cytoplasm, citrate gets converted back into acetyl-CoA and oxaloacetate, and the acetyl-CoA becomes the starting point for synthesis of cholesterol and fatty acids. These have to be tied together in a proper backbone, and glycerol provides that. So by having more pyruvate go to lactate, you could argue that ATP generation is not as efficient. In order to make up for the extra ATP that's then needed, more glucose is taken up by the cell, and we see that as a manifestation. We see that in the FDG PET scans. It's a secondary effect of the Warburg phenomenon, meaning the fate of pyruvate. And as a result of more glucose coming down, there's more left to go in both of these directions. And there's also more coming down, ultimately going to citrate, some of that coming off for the synthesis of these essential products. So that's one reason the Warburg effect is there. Now, there's some other reasons not shown on this slide. Lactate, protons come out from the cell. The acidic environment, there have been several groups suggesting that that is critical for the cancer cell's ability, in fact, to invade and to metastasize. And later I'll show you just a little bit of data that the Warburg effect, in fact, may be at the heart of why a cancer cell, or how a cancer cell, is able to evade detection by the immune system. I'll show you just an inkling of data from our lab on that. So here are the needs of a proliferating cell, and they're not always in sync. In order to get the maximum amount of ATP, you would not have invoked the Warburg effect. But then you wouldn't have the reducing equivalents that are needed for biosynthesis. So this tension exists, and there's a play back and forth between these that a cancer cell has to adjust. Well, we know a lot about the biology now of what underlies the Warburg effect. And it turns out that these two enzymes, in other words, since I'm looking at the theta pyruvate, there are two enzymes here, lactate dehydrogenase A and B. And there's an enzyme complex called the pyruvate dehydrogenase complex here. If there's less of this and more of this, I could explain the Warburg effect. Turns out that is the case in many tumor cells. What lies behind that is this factor HIF1 alpha, or the HIF group of transcription factors. So these are factors induced by hypoxia that are now well known. And one of the targets for this transcription factor family, and specifically for HIF1 alpha, is LDHA. So if HIF is upregulated, we'll come back to how that may occur in a second, LDHA goes, there's more of this enzyme made. And it also turns out that HIF, another target, is a series of enzymes called pyruvate dehydrogenase kinases. These negatively regulate this enzyme, pyruvate dehydrogenase. So more HIF means less this because of increased PDKs, and more conversion of pyruvate to lactate because of more LDHA. So then you can go back and say, well, what regulates HIF? Well, I just told you hypoxia does. But a major observation over the last 2 decades is that a number of oncogenes, when activated, and a number of tumor suppressor genes, when inactivated, lead to constitutive activation or increased amounts of HIF. And that can be at the protein level, it can be at a number of levels. So because this occurs in so many cancers, that is, a number of cancers will, of course, have mutations in these, that's the genesis of tumors, HIF is upregulated even in the absence of hypoxia, and that is why Warburg exists, or occurs. Now, there are other reasons for the Warburg effect, there are other molecular biology, which I won't get into. Now, here's a question. You would have thought that in the last 30, 40 years, somebody would have asked this question. What if I reverse the Warburg effect? Do I selectively kill cancer cells? And it wasn't asked until very, very recently. And the answer, I wouldn't be showing you these slides if the answer weren't essentially yes. So the framework is as follows. Most work is focused on genetic abnormalities, signaling changes that are downstream, and of course the phenotype that results. And all of the work that goes on in targeted therapies now is based on this framework. That is, if I intercept via targeted drugs that target specific signaling pathways, I will affect the cancer phenotype. What I'm talking about here is that more and more genetic abnormalities are being defined that then map onto specific metabolic derangements. So, for example, I haven't told you this at the moment, but it turns out that if the PI3 kinase AKT pathway is upregulated, that leads to higher levels of HIF and, of course, the Warburg effect. Now, what about this? Is this just an epiphenomenon? Or, in fact, is this arrow true? That is, if I were to block some of these metabolic derangements, reverse them, would I affect the cancer phenotype? That's the question I'm asking, in this case, with regard to the Warburg effect. Okay, so how can I do that? The good news is these are enzymes. And we know how to define, a lot of our drugs are enzyme inhibitors. Now, LDHA is a very nice target. It's a HIF-regulated gene. Turns out, getting ahead of myself a little bit, it's highly expressed, largely in muscle tissue as well as in tumors, but not many other tissues at high levels. It's in fact not expressed in red cells. B isoform is in red cells. So I could block LDHA. I could use an inhibitor of PDK, and a drug called dichloroacetate does that, and that will in fact block this pathway. And so it will in fact increase PDH and allow more to go here. I could do both of those. I could come up with other HIF inhibitors, work right at the start, if you will, and so on. Let's take a look at this. So very quickly, here is the LDHA story. It first came from Phil Leder's lab and C.V. Dang's lab. We have similar data, which we've published. It's under HIF control. It's rather tumor-specific. So what happens if you knock it down? We now have RNAi technology to enable us to answer those questions before we do any drug screens. So here's what happens to tumor volume. This is in a particular xenograft model, A549 cells. If you knock down LDHA, cells grow a lot slower. Tumors are a lot smaller than if you don't. Oxygen consumption goes up in the knocked down cells, as would be expected. You're flip-flopping the Warburg effect. So proliferation is affected, changes in metabolism, and apoptosis occurs. It turns out cleaved PARP shows up in the LDHA knockdowns, which goes away when you add N-acetylcysteine. So there's a pro-oxidant mechanism, I don't have time to get into that right now, that may underlie how these cells are getting killed by LDHA knockdown. What if you knock down PDK? What if you use dichloroacetate or use shRNAs to knock down those PDKs? This is work of Monitor et al., which really led this whole area. Really was a major resurgence in the field, this paper in Cancer Cell from the Mikolakis group in 2007. And here's what happens. They looked at these cells. You look at the apoptotic index by knocking down PDK2, and you have a vast increase in that, some decrease in the amount of proliferation of these cells. And so this is the basis for the use of dichloroacetate as a drug to reverse the Warburg effect. Now, could you exploit Warburg? Not reverse it, but utilize it. For example, could you increase intracellular acidity as a cytotoxic strategy? I'm brainstorming here. For example, could you create hyperglycemia? And if you did that at the same time as inhibiting the exit of acid or transport of lactate into the extracellular space, you might just decrease intracellular pH to the point of killing the cell. All right? You could potentially deplete ATP or decrease nucleotide or glycerol synthesis by decreasing sugar that is surrounding the cell, and other ways. So let's take a look at this. So here's a paper, 1996, not quite traditional, but over 10 years ago. Rather famous gentleman, he's still around. I actually emailed him the other day and said, did you ever follow up on this? Take a look at what he did. This is a syngeneic model, Sprague-Dawley rats, sarcomas. And he treated systemically with hyperglycemia, created hyperlactacidemia to the tune of that, right? 10 millimoles. If you go up and down a few of the stairs here in the clinical center a few times vigorously, you might get to 5, might even get to 10. It's not that wacko in terms of numbers. Okay? That's how much sugar is in the animals for 60 minutes, and he did this. And there are a number of things we can talk about that localize hyperthermia. Look what happened. About half the animals, the tumor disappeared. No follow-up on this at all. Now let's go back to the grape cure. A lot of sugar. Cells starving overnight. They're ready to go. A lot of sugar around. And what do grapes contain that might just fit the bill I just told you? Turns out grapes have a lot of interesting ingredients. And if you look in PubMed, you will see that there's an enormous amount of work going on the antioxidant activity that some of these polyphenols that are in grapes have. Resveratrol, phenolic acids, anthocyanins, some of the flavonoids. And one of those flavonoids is called quercetin. And quercetin is in the skin of the grapes. And there is very nice data that quercetin will block some of the transporters that are involved in the extrusion of lactate. MCT1, monocarboxylate transporter number 1, and others are blocked by quercetin. So is it possible that this woman, in just the right kind of patients, was giving so much sugar and yet blocking the exit of the lactate that's made that you're now dropping the intracellular pH? Could we test that today? Yes. And I'll show you how that can be done even in vivo with today's technology. So just a pure hypothesis. What about this? This will lower blood sugar, among other things. Here's one mechanism. And the anti-tumor effect would be by decreased glycolysis, ATP generation, etc. and an impact on the insulin IGF-1 axis. So this is a story in itself to how high sugars can promote insulin secretion, and in fact they can cross over to the IGF-1 receptors that are on many tumor cells. There are anti-angiogenic activities that occur when you feed somebody flaxseed oil. Flaxseed oil has a lot of linolenic acid, a lot of omega-3s, and when you look at all the various pathways and the byproducts that result, and how flaxseed oil can serve as a competitive antagonist, in effect, to that is substances made from it, to the generation, for example, of PGE2 and others. It turns out this is a simultaneous blockade of lipoxygenase, specifically lipoxygenase 5 and COX-2, for which there's lots of data in terms of angiogenesis and other events. Now, lignans are a component. They're at least the lignans that are in flaxseed oil are actually phytoestrogens. Weak estrogenic activity, but in the presence of estrogens, actually can serve as antagonists. There are not much of those lignans in the oil itself, though her crude version probably had more than the current versions of oil that you buy. But there's a lot of it in flaxseed. There's some very interesting papers that have come out from the group at Duke, a group in Canada, looking at both prostate and breast cancers respectively, where patients were fed in a pre-surgical trial, that is a 2-week window trial, fed flaxseed, and found that they had major impact on markers of proliferation and apoptosis in those cells, in the cancer cells. I should also tell you there's an NCCAM-funded study that came out earlier this year. And it's a study in chickens. The chickens were fed flaxseed. And it turns out when chickens ovulate, I didn't know this, but when start laying eggs, they have a greater tendency as they get older to develop ovarian cancer. And the severity of the disease, the lesions, the higher grade lesions, the metastases were markedly diminished in chickens who were fed this diet for a period of time. Take a look at that paper in March or May of this year. There's also an interesting relationship, and I see time's going by here, to the ketogenic diet that I don't have time to talk about. Tom Seyfried, Purna Mukherjee, and others have spent a lot of time discussing this, and there is some relationship of these two, but they're not entirely overlapping. I will leave that till later. So let me just talk about this reverse signaling issue just for a second. Turns out another metabolic derangement noted in tumors is that there's a lot of lipid synthesis. They don't like to take the lipids that are in the environment and put them into newly forming membranes. They like to synthesize their own lipids, poorly understood. And so you can ask this question: does inhibition of lipid synthesis impact tumor growth, reverse the transformed phenotype? A lot of work in this area from a number of groups. Craig Thompson, now at Memorial Sloan Kettering, has worked on this. And in fact, what he has worked on is a particular enzyme called ATP citrate lyase, which I'll come back to in just a second. So the question we're asking here is this question, this red arrow. If you were to alter these metabolic derangements, reverse them, would you affect signaling pathways? That's reverse signaling, what I'm calling reverse signaling. That's not what Thompson showed. That's what I'm going to show you. What Thompson showed was that this enzyme, ATP citrate lyase, the first enzyme in the commitment to cholesterol and fatty acid synthesis, if you were to block this, you will in fact reverse tumor genesis to some extent. Now I'm going to show you this. Quickly, a few slides that ACL inhibition signals to this pathway. So here it is. So we in fact generated cells. These are lung tumor cells. And here's what happened when you knock down ACL. They look much better behaved. They express E-cadherin, ZO1. They diminish vimentin expression. So these are now converting to a much more epithelial phenotype. That's nice. And in fact, they form smaller tumors that are more differentiated. It turns out you give a PI3 kinase inhibitor, the same kind of thing happens. They look rather similar. So we reasoned, gee, could it be that ACL inhibition actually is somewhat similar to PI3 kinase inhibition? And it turns out it is. So here's the ACL knockdown cells. You can see this is a phosphorylated version of AKT that tells you in these two residues whether in fact this pathway is activated, and it's markedly diminished, whereas the total amount of protein is the same. And because these are permanent cells, one could criticize this work and say, look, does this happen in a more immediate context, not an adjustment for long-term maintenance of these cells in culture? And so we've designed a doxycycline inducible system. As we knock down ACL with higher concentrations of doxycycline, you very dramatically knock out activation in this pathway. And this is phospho-ERK, another important pathway, of course. Here's E-cadherin going up and a repressor of E-cadherin going down. So here's an example, and there'll be many to follow, where by acting on metabolic pathways, we can affect canonical signaling pathways that the world in general loves so much. Okay, let me skip this here. Tumor immunology in 5 slides or less. So this is a synthesis of an enormous amount of work, and I'm only citing 2 authors here, Laurent Zitvogel and Polly Matzinger, who's here at the NIH. In order to get effective immunotherapy, you need to at least do 3 things. You need to reverse the immunosuppressive environment. You need to make sure that antigen is appropriately presented to effector cells, and you need to get these effector cells really activated. You also need to start and maintain this immune response. It tends to shut off very rapidly. You need to figure out what the type of that response is. The type of immunoglobulin, the type of T cells, et cetera, et cetera. Or do you want T cells versus meaning adaptive versus, for example, the innate response? And it's got to get. Location is everything, as it is in real estate. You've got to get the effector response to where you want it. So, again, not a lot of time here, but basically there are several mechanisms by which tumors tend to create around themselves and in the host an immunosuppressive environment. 3 cellular mechanisms are the generation of so-called T regulatory cells, these so-called myeloid-derived suppressor cells, and these so-called tumor-associated macrophages that are polarized in the M2 direction. My purpose of telling you this is that I'm going to show you how a dietary manipulation can actually affect these in just a few minutes. So first of all, let's go back to Coley. How does Coley measure up? The science. There's no doubt that there are many products in this mixture. There's exotoxin, there's endotoxin. He's got a gram positive or gram negative. And these are going to activate all kinds of interesting so-called toll-like receptors, as well as do many other things. And that component is really in that second, third bracket that I just showed you. You're going to change the local environment. So the dendritic cells now get excited. The local monocytes macrophages may turn more towards an M1 phenotype, and so on and so forth. And these dendritic cells will then gobble up the antigen, go to lymph nodes, et cetera, et cetera. That part's pretty strong. Probably the innate system is what's being invoked here, because the action as seen in patients is fairly quick. This is not to discount that there's some aspect of adaptive immunity, and again, we can spend some time here. Could you help Coley out by actually, now that we know more about the immune system, by pre-treating, and there are drugs around that can do this, that will deplete Tregs and MDSCs before giving Coley? Is it that we're not presenting antigen just quite right? A short burst of radiation or short amount of chemotherapy, not to kill all tumor cells, but just enough? Or vaccination just at the right time for better antigen presentation? And most importantly, we must monitor what we're doing, which we can now do by looking at a whole bunch of downstream consequences. Is this only immunotherapy? What about the fever? Fascinating data on how fever activates the immune system in a whole number of ways. Other things. Turns out there are plasminogen activators, streptokinase, and a whole bunch of other crazy molecules of that sort that are in this bacterial mix. Could they be disrupting the fibrin clots that surround tumors, fibrin matrix that surround tumors, and better have the immune system come in? Speculative ideas. But here are the challenges. The way that Coley's could move into the clinic may require a processing that may destroy its activity. Again, we can talk more about this. No single mechanism of action. So in today's molecular reductionistic world, this often falls between the cracks. And then there's this issue. There is no intellectual property. A company in Canada tried to patent the process. The process has been on the web for the last 50 years. They didn't succeed. No investors want to put money into a company that's investing in Coley's. Okay, so here's just a little bit about these two together. So these MDSCs, isolated from melanoma tumors, if we reduce LDHA, we're reversing Warburg. Here's what happens to these MDSCs. They show up in this right quadrant based on these 2 markers, way down if you reverse the Warburg effect. Okay? So this is the intersection of metabolism with immunology. A ketogenic diet, which I know we haven't spent a lot of time on, looking again at these myeloid suppressors, they go from about 6% to about 0.34%. This is all in mice in a pancreatic tumor model. And these infamous T regulatory cells, on which there are hundreds of papers now out, a short course of a ketogenic diet will bring these back essentially to normal. So here are dietary manipulations affecting metabolism that have direct impact on what's going on in the tumor microenvironment. So the last part of this talk, am I supposed to end at 9:45 or go about 9? Until 10? Okay, I'll try to finish before 5 or 10. Okay, terrific, thank you. Sorry, so let me then go to part number 3, biomarkers. So biomarkers are particularly important when we're talking about therapies, and in many of these traditional therapies, as well as others that we're more used to thinking about, herbal products, for example. There clearly are going to be multiple mechanisms of action at play. And in some cases, there will be multiple active and uncharacterized agents in the mix. And so particularly in this situation, it's going to be important to think about biomarkers in the trials that we do. Farnsworth was one of the speakers here about 5-6 years ago. And he pointed out that despite that it's very important to have biochemical standardization in the way these studies are done, for example, where herbal products might be involved. It'll also be important to look at some in vitro biological standardization, that is, some in vitro evidence of activity. And these then can be parlayed potentially into in vivo biomarker studies. Now I'm going to show you that in just a second as to how one example of that. So here it is, back to metabolism. Can we actually visualize the change in fate of pyruvate in vivo? So if we were to take DCA to the clinic, as it is now being taken, or if we ever wanted to investigate a version of the Grape cure by creating hyperglycemia and blocking the extrusion of lactate, can we provide evidence, in fact, that in that tumor, in vivo, in humans, we are changing the fate of pyruvate? Why is that important? Let's think about it. If there's efficacy in the treatment, and if that correlates with what we think we are hitting as a target, meaning the fate of pyruvate, that's tremendous. In fact, in later studies, we can use that initial as a surrogate for what we think will be the final outcome, and saving much time and cost. But what if the treatment doesn't work? But what if a target doesn't get hit? Well, then maybe the scheduling is wrong, or the dosing is wrong, etc. If the therapy doesn't work, and the target is hit, bad news. If the therapy works, and the target is not hit, something else is happening. So we learn something very important. So can we do this? Can we non-invasively, in fact, find out? So here's a little machine. I go back to my physics days here. So this machine, I'm telling you about MRI. Conventional MRI, there are thermal fluctuations that randomize the nuclei, and so you get low magnetization. But if you cool to very low temperature, the nuclei can be aligned. And this technology is called hyperpolarizing MRI. It's an MR spectroscopic method. A number of agents have been used, and this is the one that we've been using at our institution. So that carbon-13 then is the tip-off. And you can now look at the signature over time by looking at MR signal versus frequency. You can start seeing the pyruvate peak coming up as you focus the coils over the tumor or the muscle in question, and you start seeing the other peaks, alanine and lactate and hydrated pyruvate, and then it goes away. So here's what happens. You take an animal bearing a tumor and you give that animal DCA. Here's pyruvate, here's the lactate coming up as a function of time. This is seconds. When you give the DCA, you'll see a lot less of the lactate come up. So in vivo now, you have made a change in the fate of pyruvate. And this can be scaled to humans. This technology is ready to go in man and will be there in a few years from now. And so we will be able to observe these metabolic pathways in vivo by the right kind of labeling agents. So another big use, actually in the interest of time, let me move ahead here, of these biomarkers is when there is conflicting data. And I can't give a talk here, sponsored by NC Camp, without talking about stress. There's a fascinating series of observations on stress and cancer. And I'm just going to focus on stress and cancer progression. So this is a slide from some of Sood's work, Anil Sood. And this is from a particular review, pointing out that a number of stressors and psychological processes lead to at least two major outputs from the brain. There is the so-called hypothalamic pituitary adrenal axis, powered up by ACTH, etc. And then there's the autonomic nervous system. And he has tended to focus his attention over here, and a whole number of downstream events occur. There are immune and stromal cell effects, decreased NK cell function, decreased T cell activity, increased Tregs, increased HAMs. You've just heard all of these from me a few minutes ago. Cancer cells migrate, invade better. There's more angiogenesis. And he's focused on this. I'm just going to take you through one slide of his. And that is that epinephrine and norepinephrine, through the beta-2 adrenergic receptor on cancer cells. Turns out some cancer cells have these receptors. Turns on VEGF production by the cancer cells, a pro-angiogenic molecule, vascular endothelial growth factor. And that increases tumor growth and increases vascularity. Key paper, this is a review actually, the paper was in Nature Medicine a year before. And so giving the drug propranolol, which of course will block both beta-1 and beta-2, will in fact reverse this angiogenic response. So in his animals, these are ovarian cancer-bearing tumors, animals, he stresses the animals by a confinement every day for a period of about a half hour. Tumors grow about twice as fast, and they revert back to that original state of growth if given a beta blocker. Okay? Sounds kind of interesting. Totally contradictory data. Came out this year. In fact, they don't reference Sood's paper. It's kind of interesting. I'm an outsider to the field, so I'm watching this. This paper from a Canadian group found that if you put animals bearing tumors in a so-called EE, an environmentally enriched place. And if you look at the movie on the web, you'll see the cages. The animals can find all kinds of places to hide around and play around and this and that. They are very engaged. Very different from being restrained. In fact, quite the opposite. And it turns out, and that's why this paper is in Cell, that they figured out why, under that environment, tumors actually grow a lot slower. And it turns out through a series of very elegant studies, these animals turn out from their hypothalamus to make this factor, this brain-derived neurotropic factor. And it signals into the fat cells, of all things, through the sympathetic system, and through the beta-adrenergic signaling, it turns out, to decrease the production of leptin. And this pathway they mapped out. For example, they took an animal without putting the animal in an environmental enriched place. They simply gave that animal the BDNF, and the whole thing happened. Now here's the problem. If you now put propranolol in this context, tumors grow faster. The exact opposite. So how do you resolve this? Do you do a clinical trial on beta blockers or do you not? And I suggest to you that there are many other things beta blockers will do. And the point is, there are a whole number of downstream markers one should probably look at. In the interest of time, I'm not going to spend more time on this, but the idea is we should look at the immunologic axes. We should look at various metabolic axes, the angiogenic axis, in patients if we're going to engage in such a trial to get anything meaningful out of what we might do. Okay, last part. In the last three sentences of this paper you have seen already, here's the comment. This study represents an approach to efficient tumor therapy with a potential application in clinical oncology in the not-too-distant future. Well, no clinical protocol has been done with this. I think you all know by now the reasons why. Nobody's going to make a lot of money. Here's a person who was very dear to me. Passed away a few days ago. His family may be in the audience and has given me permission to mention this case. Metastatic colorectal cancer, standard of care exhausted. He requested a cocktail, he was a biochemist, of available drugs based on chemosensitivity testing of his tumor, a controversial topic in oncology. Request was denied at 3 medical institutions. He then ended up being the first U.S. patient under an emergency IND to receive inpatient Coley's toxin. Unfortunately, he passed away a few days afterwards. After that approval was granted, he was given 5 doses, but to no avail. You'll see in a second why I'm telling you this. So you've heard this from me. And I think most of you will probably buy into this, that there exist therapies that are potentially promising. Perhaps some of the ones I've shown you, and many, many others that are not part of our mainstream medicine. Drug development is expensive, it's inefficient, it's lengthy, so typically it's undertaken by pharma. And pharma is not going to do this unless they can recover the costs. This is their obligation to their shareholders. There is nothing wrong about this. So they must look for a patent on the drug. They've got to look at market size. They've got to ask, is the biology well enough known that the chances of success are reasonable? You don't see that many drugs in sepsis, for example, because it's a tough topic from a biology standpoint. And if the estimated return on the investment is low, it is dropped. I refer to those entities, those ideas, those therapies as fiscal orphans. And ultimately, it's the patients that lose. So what are examples? Categories of FDA-approved drugs that could be used for generic FDA-approved drugs that could be used for new use. Even if a patent, first of all, a patent may not be obtainable because of prior disclosure, but even if it is, it may be hard to enforce, or the price point may be very different. A statin for cancer, even if you could get a patent on that, you would have to price it very differently, which you can't, because the cardiovascular market is so huge. Substances never patented, vitamins, certain herbal products, therapies such as lifestyle changes, diet, as we've talked about, and other non-patentable therapies. The pity is, actually, these are ready to go. There's nothing stopping anybody from actually doing rigorous trials with these. And in fact, a recent initiative now from Francis Collins is suggestive that this institute, there may be a program within it, the Cures Acceleration Network, that part of which may be to incentivize the development of these ideas. At the end of the day, it has to be the public. So my wife, who's in the audience, Vidula, and I decided to get going in our own little way. So we formed this organization about 3 years ago. And the idea is to do what I just told you, to look for promising therapies that are implementable, which the for-profit sector is shunning because the fiscal returns are just not there. So the idea is that we will enable patients to find such therapies, share their experiences, and I want to spend a minute on that, and fund, ideally, this research. The first step is, of course, to find promising ideas. And we expect the world to come to us and back and forth. We are inviting ideas from patients, from many other sources. This is what we ultimately want to do. We want to conceive, and the we in this case is a group of individuals, the larger the better, to actually write and ultimately sponsor these trials. But before that, there is another alternative. It's not to replace number 3, but it's to ask whether in today's internet era we can actually collect useful information from those already using off-label drugs. So this last 4 slides, what about this? What about patients such as FS that I just presented to you? This is the dilemma that we face. In certain cases, he was asking for off-label use of available drugs, but there were no trials. There was data, some in fact even in humans. But there were no phase 3 data, the hallmark of what most physicians would want to do. In other cases, he was asking for therapies like Coley's as a last desperate effort, if you will, where they were never approved for human use for any indication. So is it time to think about this again? This is what the law states about off-label use, that under certain circumstances, physicians in fact have the right, in fact in some ways, are almost obligated to think about off-label use if there is this kind of data already there. But in reality, it doesn't happen. It doesn't happen. It happens somewhat in different disease states, but in other cases it does not at all, because physicians worry about all these things. For example, how do they know what to monitor? Not at all clear. This whole issue, therapies never approved, is it time to re-examine this issue of access for patients with a fatal illness? Is it time to think about a terminal patient's Bill of Rights? But there are some real issues here. How do you protect patients from quack medicine? How do you protect physicians from lawsuits? Could this be used by drug companies in an inappropriate fashion to evade or to bypass phase 1 studies in a proper manner? There's a lot to think about. So let me conclude then with this summary slide. I've tried to show you that new insights into what may seem outlandish ideas may come from these two areas as far as cancer and are worth thinking about. Biomarkers have a major role to play in moving such therapies forward. And just because a therapy is not part of mainstream medicine does not mean that it is ineffective, and that we need the public ultimately to support this effort. So I'll leave this last slide, or last two slides here with you. We must incentivize the testing and development of promising ideas, no matter how out of the box they are, or whether they will make money or not. And priority should be on what we can test immediately. Thanks to all the various people in my lab. Some of the data that I've shown you was generated by them. Pankaj is now setting up his own lab and has done a lot of the LDHA work. And these are the folks in our radiology department who have helped with the hyperpolarizer work. And my wife is the one who keeps me honest. Thank you.
We have a few minutes for questions for Dr. Sukhatme. Yes, Anne.
I'm Ann Ashby from the Foundation for the NIH, and I'd be interested to know if other foundations like the American Cancer Society are interested in partnering with you and maybe funding global tours. Where are you going to seek support?
Well, so we have begun on that route. We would very much want to work with whoever wants to work with us. We have had a brief discussion with the ACS. There are some complexities. I guess I can put it to you that way, or we can talk a little bit later. But we would very much like to work with anybody who, in any sort of partnership, who feels that these are worthy goals to pursue.
The MRI-based method of assessing pyruvate lactate ratios was fascinating to me. What will be the steps to move? I gather those are rodent studies, or
those are rodent studies, but those are rodent studies, Josie. But hyperpolarized. So basically, the technology approval in humans is not there yet, but it should be. There are clear ways, because it's an MR spectroscopy. It's an MR-based method, basically. The issues of how much pyruvate you're injecting in and so on, and the toxicities that one may get from that, all will have to be assessed. So every agent would have to be approved in its own way. But from a technology standpoint and from the standpoint of science, because it is MR technology, it is ready to go in humans.
Vikas, beautiful talk. Is it likely that you're going to end up with, you're talking about combinations of biomarkers or panel of biomarkers. You'll probably also be working with a panel of drugs using sort of combination chemotherapy here. The FDA is in the process of coming up with some guidance on how to do that, but it sounds like the biomarker may actually help you. It may help tell you this part is whether things are additive or super additive, I guess.
No,
have you given any thought to how to get these kinds of things through the FDA, I guess is the question.
Yeah, no, well, you raise an important issue, and the idea, or the question of how you move combinations forward is not a simple one. So again, I think panels, especially panels that cross different axes in terms of mechanisms, immunomonitoring, metabolic monitoring, angiogenic monitoring, among others, I think will be very useful in this. And I think there'll be a discovery sort of component of these, where there'll be a limited number of studies done with a larger number of these biomarkers, or markers, if you will. And then one could hone that down in a proper, at the time of a more, time of a larger trial. So it may actually work in two steps. It may be sort of a more pilot phase, if you will. And then depending on how things sort themselves out, then one can incorporate a more limited number of these in larger studies. I don't know if that quite answers the question. There is no simple answer to this question.
Can we use the microphones in the aisles for questions, please?
Can we ask you to go to the mic?
I'm Steve Straus' brother. When you're in the trenches and you have an oncology patient who has failed and you're looking to individualize therapy, there's an enormous barrier with third-party reimbursement. You know, if you want to put together some novel combination, the chances certain very acceptable drugs will even be approved is close to zero. And, you know, this is a barrier that you're going to face with almost anything. People are just not getting access to many of the drugs.
You're absolutely correct. You're absolutely right. I guess one way to turn that around, two comments. One is that one of the reasons one wants to do rigorous trials is in fact to be able to then publish peer-reviewed data that would then make the insurance companies, I shouldn't say make, but suggest to the insurance companies that they ought to now cover that therapy. So in fact, that is a very important rationale for in fact engaging in the trials, not just for the science, of course, and hopefully helping people, but to make sure that those then are acceptable for insurance reimbursement. I will tell you, though, that we are finding perhaps a small number of patients who are willing to pay on their own, and it may be that those initial reports that emanate from that group of people will still be of enough guidance that then others will follow, trials will occur, and so on and so forth. So they may form the seed, but you're absolutely right. This is a major, major issue.
Well, I have a patient who has an extremely rare cancer, an older teenager. There's been almost no reported responses in the literature. And I went in and asked for approval of 5-FU because it cost about $1 a day. And the insurance company approved it. And then I asked for what I really wanted, and they turned down everything. And I asked the basis for approving the 5-FU. Of course, there was no basis. I think we finally got approval. It took a lot of work because there was nothing reported that ever worked. This is going to be a tremendous dilemma, and especially when you have anything novel or rare diseases.
Thank you, Mark, and thank you, Vikas, for a wonderful talk. I think Vikas articulated very well the promise of modern biomarker array, complex omic kind of approaches to begin to allow us to pull apart the potential synergisms and interactions in some of these traditional remedies. Jeff White from the Cancer Institute is here, and I know they struggle, as do we, with methodology and how to get definitive answers about complex interventions. So thank you, Vikas, for a very provocative and thoughtful kind of talk. Let me tell you about the next steps in our NCAM celebration day. We do have a reception outside. I do want to thank the foundation of the NIH and Bernard and Barbara Osher, who made today's celebrations possible. We have invited 5 NCAM grantees to be with us here today and to participate in a poster session that will begin after we've all had a chance to get a cup of coffee. I do want to introduce our visitors and hope that you will look at their interesting work, which we were lucky to be able to support with the ARA stimulus funds. And I will ask our 5 visitors to stand. Guido Pauli is working on the validation by spectrophometric methods of botanical products. William Lafferty is bringing comparative effectiveness research to complementary and alternative medicine in his assessment of methods to look at CAM and its impact on health costs and utilization. Giresh Shekhara is doing very innovative studies on probiotics. Adam Perlman is undertaking a valuable study of massage, and George Salem a study of yoga. So I think their work indicates the breadth of the kind of science that NCAM is supporting currently. I think you'll find their presentations very interesting. So thank you all for your presence and your attention, and we look forward to talking with you at the reception.